Observe the human central nervous system. Tucked beneath the cerebral hemispheres lies a distinct, intricately folded structure, the cerebellum. By physical volume, the cerebellum is modest, accounting for roughly 10% of the brain's total mass. Yet that small footprint houses a biological paradox. It contains more than half of the total neurons in the entire central nervous system. This extreme neural density does not translate into motor initiation. The cerebellum does not actually trigger voluntary movement. Its immense processing power is instead dedicated to regulation and integration, managing the complex timing of signals as they travel through the nervous system. The cerebellum functions as a high-density cybernetic processing center, providing the integration required to enrich the quality of every movement. When this processing hub is damaged, the result is cerebellar ataxia, a term that literally translates to not ordered. A patient with ataxia retains their physical strength and the basic ability to execute a voluntary movement. The impairment lies in the smooth coordination, the timing, and the rhythm of those actions. Clinically, this manifests as tremors, generalized unsteadiness, and delayed response times. To understand these symptoms, we have to look past the visible clumsiness and identify the specific mechanical failures within the brain's circuitry. Ataxia represents the measurable result of a specific breakdown in the brain's internal integration and feedback mechanisms. This schematic details the cerebellum's structural wiring, identifying the cortex and deep cerebellar nuclei. The system receives inputs from the cerebral cortex and sends signals back through the deep nuclei. At the base, the vestibulocerebellum modulates postural balance and eye movements. Damage to this specific pathway results in nystagmus, rapid, involuntary eye movements. Running down the center is the spinocerebellum. It modulates muscle tone and posture by processing signals through the fastigial and interposed nuclei. The lateral hemispheres form the cerebrocerebellum. This zone regulates fine motor planning, outputting via the dentate nucleus. One definitive diagnostic rule for these pathways is that unilateral cerebellar lesions affect the ipsilateral, or same, side of the body. Because the cerebellum is highly compartmentalized, regional damage can selectively destroy balance, tone, or fine motor skills independently. Beyond its anatomical map, the cerebellum functions as an active feedback loop that regulates actions in progress. The cerebellum receives a copy of the intended motor plan directly from the cerebral cortex. Simultaneously, it receives real-time sensory data from the body. It acts as a comparator, instantly calculating the deviation to output a correction signal. This high-speed loop allows for microcorrections during a movement, often before an error becomes visible to the eye. If a lesion severs this loop, the brain cannot perceive its own physical errors, resulting in the uncorrected overshoot seen in ataxia. We can apply this framework to specific clinical presentations, starting with a failure of the spinocerebellum, hypotonia. Hypotonia is caused by a decrease in excitation signals from the deep cerebellar nuclei to the alpha and gamma motor neurons. During an exam, the muscle feels abnormally heavy and lacks firmness upon palpation. This loss of underlying tone is visible during the pendular knee-jerk test, where the leg swings purely by its own weight and momentum. When the lateral cerebrocerebellum fails, it results in dyssynergia, also known as movement decomposition. In this state, the brain fails to properly sequence synergic muscle groups, breaking fluid motions into disjointed, robotic steps. During the heel-shin test, the patient's leg exhibits jerky, erratic targeting rather than a smooth slide. These presentations demonstrate that baseline muscle firmness and smooth sequencing are active, continuous neurological calculations. The failure of the agonist-antagonist muscle relationship is best seen in the rebound phenomenon. A patient pushes hard against a therapist's resistance. When that resistance is suddenly released, the result is violent. The patient's arm overshoots because the cerebellum fails to trigger the antagonist muscle in time to break the motion. This impairment in the timing of muscle force is known as dysmetria, causing inaccurate movement amplitudes. This includes hypermetria, an excessive overshoot during fast movements, and hypometria, where the movement falls short of its target. Cerebellar tremors, specifically intention or terminal tremors, are the visible oscillation of these late, miscalculated force corrections happening around a joint. These isolated failures coalesce into the hallmark clinical sign, the ataxic gait. The widened base of support provides structural compensation for vestibulocerebellar unsteadiness. The irregular stepping direction and reduced stride length are manifestations of spinocerebellar and cerebrocerebellar timing errors. Performing a tandem walk, heel to toe, acts as the ultimate clinical stress test, requiring peak integration from all cerebellar zones simultaneously. Because this test demands total integration, it serves as the standard metric for tracking a patient's rehabilitation progress. The cerebellum functions as the movement regulatory center, constructing synergy throughout the duration of every action. When it fails, the capacity for movement remains, but human motion devolves into a cascade of uncorrected, raw physical forces.